Alright folks, I thought I'd put together a quick little video here to talk about diffusion and osmosis. This is probably one of the most important concepts, I would say, in this course, really, because the idea of diffusion and osmosis really is central to understanding how a lot of things move in the body. So for instance, how does oxygen move from the air into your tissues ultimately, or how do your kidneys function, or how is it that we're able to move substances from our digestive system into our bloodstream. Understanding the basics of what is diffusion and osmosis really is the foundation to all of that. And so if you understand generally what that is, then understanding those different concepts isn't quite as tough. So let me just go in here and do some drawings. First, when we're talking about like diffusion and osmosis, one of the things we need to do is kind of define some terms, right? So if we're talking about, hypotonic, I said one thing, I meant another. Hypertonic, hypotonic, and isotonic solutions. These three terms are comparative terms. What do I mean by that? Well, if I just said this stylus, right? If I just said this stylus is big, well, compared to what, right? I mean, compared to my car, it's not big. Compared to an ant, it is, right? And so these three terms are comparative, and you compare them based on the concentration of a solution, right? And so that means we need to define what a solution is, right? So a solution is a solute plus a solvent. A lot of times students think of solvents as, maybe you're thinking of an organic solvent, like, I don't know, acetone or something. But a solvent can be any kind of liquid that's dissolving some other thing, right? Most of the time solvents in biology is water. Our solutes would be things like maybe salts or sugars, right? All those things would be solutes. So anything that's dissolved in a substance would be the solute. And we typically name solutions based on the solute that is there. So, right, if I said a 10% salt solution, well, your assumption is it's 10% salt and 90% water. If I'm in chemistry, I might say 10% salt in 75% alcohol, right? ETOH, right? And in that situation, you would define what the solvent is. But if you just say 10% salt solution, kind of the understanding there is that the solvent is water, right? And so now let's define what these terms are. So let me move this up. If I'm talking about a solution that is hypertonic, right? What that means is a hypertonic solution is a solution that has a higher solute concentration compared to another solution, right? So a higher solute concentration compared to another solution. Not compared to the solvent, not compared to itself, right? Compared to another solution. So then you can probably assume that a hypotonic solution has a lower solute concentration, and then an isotonic solution, the solute concentration of the two is equal. So just being consistent, I'll draw up and down. And so then if we look at that in real world, right? So let's say I have solution A, right? And it is 10%, whatever. It could be salt, sugar, it doesn't matter. And I have solution B, right? And it is 20%. Again, it has to be the same thing. So these would be salt solutions. Solution B would be hypertonic because it has a higher solute concentration. Solution A would be hypotonic because it has a lower solute concentration. Right? Well, what happens then? Let's say we have a larger container, right? And in that larger container, let's say we have a membrane, right? right? And that membrane is permeable to water and water only, right? So we would say it's semi-permeable, and it's semi-permeable to water. Well then, let's say if I move these solutions down, so now I've got solution A over here and solution B over there, right? I might want to ask you, in which direction would water move, from A to B or B to A? Well, what you have to remember about movements of substances is they're going to move down their own concentration gradient, right? So up here, when I defined my solutions, I said 10% salt and 10% salt. Well, what that means then is, if it's 10% salt, then it's 90% water. If it's 20% salt, then it's 80% water. And since solutions move down their own concentration gradients, right, what that would mean is that this is 80% water, this is 90% water. So if this membrane is permeable to water, water is going to move from high concentration of water to low concentration of water, right? So if the solution started out at this level, equal levels, right, what's going to happen is this level is going to drop while this level increases, right? Because water is going to move from high concentration of water to low concentration of water, right? And it's going to do that passively. It doesn't require any energy to do that. It's just through osmosis, right? So osmosis is the movement of water across a semi-permeable membrane. Okay. Now, the other thing that could happen here, so if the membrane was also permeable to salt, right? So if I go back and let me just erase these, right? And we'll start with our water levels, the same again. So if this membrane was permeable to both water and salt, right? Well, what would happen is, since I would have 20% salt over here and 10% here, the salt would move in this direction, right? So I'm keeping the salt, the black color, right? So salt would move from high concentration of salt to low concentration of salt. Water, however, would move from high concentration of water to low concentration of water, right? And so all molecules will diffuse down their own concentration gradient. So really, diffusion is the movement of any molecule down a concentration gradient. Osmosis is specifically the movement of water down its concentration gradient, right? And both of these processes are passive, meaning they do not require energy for them to occur. They will occur spontaneously, right? Now, why would all this matter? So we were talking about this kind of outside the body, and this is an anatomy class, so let's talk about what would happen inside the body, right? So let's say, we can just keep this blue here if we're going to talk about IV fluids. So let's say we've got a vein here, right? And inside that vein, let's say we've got a red blood cell, right? Now, let's say we take a needle, and we stick a needle, right, in that vein, and we inject pure water into that vein. If it's pure water, it's 100% water. Now, you might not know what the concentration of solutes are in a red blood cell, but you know inside a red blood cell isn't pure water, right? So you don't really need to know what the concentration is inside the cell, right? You would be able to tell me what's going to happen to this red blood cell. Well, because it's not pure water inside, it's not 100% water. That means water must travel, this water that you're injecting must travel into that red blood cell, right? Because you have 100% water out here and less than 100% in there, right? So when it's traveling into that red blood cell is going to swell, and eventually it's going to lyse, something called hemolysis, right, or a hemolytic process. And when it lyses, right, it's just going to have these, it's going to put these holes in it, and all the contents of the red blood cell is going to come out, right? And that is not a good day for our kidneys and everything else, right? And so that's what's going to happen. Let's say, though, instead of 100% water, let's say maybe what you injected, or for whatever reason, let's say it was a, I don't know, let's say it was a 20% salt solution that you injected in here. Well, now what's going to happen, instead of this red blood cell pulling water in, right, water's going to leave that red blood cell, right, and it's going to undergo a process called crenation, right? It's going to shrivel, and it's not going to want to travel through your veins and arteries the way it should. Basically, you're dehydrating it when you do that, right? And so there would be all sorts of reasons that you might want to give somebody fluids. Typically, you would want to give somebody fluids if they are dehydrated. Sometimes if your blood pressure drops too low, especially if you're in a hospital environment, they can give you fluids to help increase your blood pressure. And again, in that case, that's why you wouldn't necessarily want to give them pure water, right? Because the pure water in that case, right? So if I go back to my 100% water situation, right? If you injected that pure water is definitely hypotonic to the red blood cell, right? It would be hypotonic to everything in there. The red blood cell would be hypertonic, right? And water will always, if there's a semi-permeable membrane, water will always move from hypo to hyper, right? And I hope that makes some sense. Now, let me just talk about some pitfalls here that students usually make. If I go back up here and I talk about hyper, hypo, and isotonic, if I were to ask you to define hypertonic, what is a hypertonic solution? Sometimes what students will tell me is a hypertonic solution is a solution that has a higher solute, what they'll say is a hypertonic solution is a solution that has more solute than another one. I would be nitpicking here a little bit, and I get it, but I want to talk about why that's wrong. If you just say more, right, you're not really talking about the concentration. More, not necessarily, right? So, for instance, let's say I had two, wow, my pad moved here. Let's say I had two solutions, again, right? And so here I've got solution A, and here I've got solution B. And the difference is the volumes. So let's say solution A is, let's say it's 1,000,000 gallons. And it's 1,000,000 gallons of a 0.01% salt. Right? And then let's say solution B is, well, let's change the units here. Let's say this is 1,000 liters, right? Just to keep it consistent. Right? And then let's say over here I've got 0.5 milliliters of a 10% salt solution. Right? Well, there's 1,000 milliliters in a liter, so that gives you an idea for how much bigger this is. If I just said that hypertonic was based on more solute, A would have more solute in it, so if you counted up the individual salt particles in solution, A would have more than B, just based on volumes. But A is still hypotonic and B is still hypertonic because you're worried about the concentrations of solutions and not just whether there's more solute, right? And I hope that makes some sense there, right? And I realize sometimes students think that's nitpicking a little bit, but terminology matters. What you say matters. So a lot of you that, maybe you're going into EMT or EMS or nursing, or it seems like what other programs take this, radiography, right? An important thing is making sure that your terminology, you're saying what you should be saying or want to say. Sometimes you can see this in patients. Maybe they leave a room and they're repeating what someone says. If what you tell them is not accurate, then that can be a problem, right? Or what you tell a doctor or what you report is not accurate, that can be a problem. So you have to be really diligent about the terms that you use. So saying more solute, again, technically not correct there. The other one that's obviously wrong is sometimes I'll get a student that says a hypertonic solution has more solute than solvent, right? Now, you can't compare the solution to itself. You have to compare a solution to something else. So saying a hypertonic solution has more solute than solvent, that's just not correct, right? So be careful with those. I hope that makes some sense. I hope you can apply that. One of the things that you should always think about in this course is anytime there's some concept like tonicity, you should think about that in terms of the human body as best you can. And that's one of the reasons that I try to use examples like these. All right. I hope that helps. If you have any questions, let me know.